next presentation will be by Dr. John Zaia from City of Hope, and he'll talk about the use of zinc finger nucleases to modify hematopoietic stem cells in HIV-infected subjects. Thank you very much, John, for coming.
Thank you, and I want to thank the organizers for inviting me. This is the first clinical trial using genome editing of autologous hematopoietic cells in anyone, actually, but especially in persons living with HIV. So it's a very exciting study. I can only show you the preliminary results that we have to date, but hopefully by the time of CROI next year, we'll have all the data we need. So you've heard the idea. You want to take a donor. You can do it with an allogeneic setting, like we've heard. Or what about taking the person himself, the autologous setting, and just genetically modify those? So what we're seeing here is the various mechanisms that we have now to isolate the cells. It's a Clinimax unit that's made by Molteni, and then put them through an electroporator. This is an electrical device, and you put into the device messenger RNA that encodes the nuclease that you want to use for the genome editing, in this case, zinc finger nuclease. And you zap the cells, and it's about 1 billion cells, and no one had ever figured out how you take 1 billion cells and put electrical current through them, and then get those cells to engraft and express in the progeny the genome edited effect. So that's what we're going to show you, and that's I think the importance of this study. Now it all arose, of course, the question then, is this safe, and is it really feasible using zinc finger technology? And then one of the key take-home messages that I'm going to tell you, we do think it is safe and feasible, and I'll show you the data. The technology, though, is evolving, and you'll see some weaknesses of the current method. For cure strategy, it's not yet ready, but it is a start. So it all began really with Paula Cannon's work. She was working with people from Sangamo who had really developed zinc finger nuclease. And a zinc finger nuclease is a synthetic protein that has fingers on it. These are peptides. And these peptides can bind to specific sequences of a gene. And empirically, you can figure out which peptides you need for which gene. So they created one for CCR5 and attached to it a cutting enzyme called FOC1. And that's an enzyme that is a nuclease that can do a double-strand break on DNA. So this molecule then can wrap around the specific DNA at the site of the CCR5 coding region, can break it in a double-strand break, and then you can then heal that break with the NHEJ, the non-homologous end joining, and you disrupt the reading frame at that point, which is exactly what we want. So these cells will have in their gene now a disrupted CCR5, and it will not be expressed. Of course, each cell has two, so you have to do it to both of them. And in fact, in the system, you don't always get homologous disruption. You may only get heterologous disruption. And this is shown here. So you can see here in an old slide that was done, I think probably by Paula or by Sangamo. And in any event, you can see that the alleles are encoding the CCR5 are disrupted here, at least 25%. Now this is what I call the Model A forward zinc finger nuclease. They have been improved since that time. You can get up to 75, 80, 85%. And some of the new zinc finger that are in clinical trial for thalassemia, for example, by Sangamo, are much more efficient. But this is what we had, and this is what we started with. It targets CCR5 at this site right here. This is Timothy Brown's CCR5, and what Monique has described in the cord blood bank. This is where the disruption occurs. But here, the disruption occurs here, and all of the reading frame is disrupted. So this is the data from Paula's lab that Nat Holt published with her in 2010. And it showed that, in fact, the system worked very nicely in mice. And these mice were transplanted. Then when they engrafted, they were exposed to HIV. Then they grew HIV. But for about 6 weeks, they looked like the controls. And they made good HIV. And then the HIV deteriorated and went down to undetectable levels, while the controls continued to express HIV. And what was happening at this time was the CD4 cells were being selected for the CCR5 negative cells. So you got eventually up to about 50, 60, 70% of those cells, of the CD4 cells in the mice, were in fact CCR5 negative. So this gave us the idea, why don't we do a clinical trial? We'll see if we can engraft patients with these cells, and then we'll remove the heart therapy and during an ATI, determine whether you can actually select for these resistant cells. So that's the trial that I'm going to describe now. The PI is Marina Krishnan. She's a well-known person in the field of transplantation, had done many transplants autologously for AIDS lymphoma, and she's led the team at City of Hope. Phil Gregory at Sangamo, Mike Holmes, Dale Ando, Ed Connor, and now Adrian Wolfson, a series of people that have come through Sangamo, were our co-workers at Sangamo, and they co-funded this with CIRM. And the CIRM team was Sohail Talib, and he brought in David Ho as his advisor, and Lee Jewell was the community advisor. So they worked with us on setting up this trial. And it was done at 3 major sites. Tony Mills' site in Los Angeles enrolled patients, Jay Lalazari in San Francisco, and Ron Masiasu at UCLA. And the goal was to treat 9 patients. This slide is busy, but let me just run it through you quickly. You had to have R5 virus. You had to have no detectable HIV, and you had to have CD4 counts between 200 and 750. And this is the product, the autologous product that was made by mobilizing your cells out of your marrow with GCSF and Plerixafor. We then electroporated messenger RNA into those cells, and we gave you back at least 2 million cells per kilogram. This was after busulfan conditioning, and we gave 2 doses. In cohort 1, they got a relatively low dose, 8,000, we call AUC, area under the curve, is the exposure level of busulfan. Cohort 2 went up to 12,000. Now, put this into perspective. For hemoglobinopathies, thalassemia, sickle cell disease, autologous transplants for gene therapy use 20,000, approximately 20 to 22,000 AUC. So we're a little less than half the totally myelo-depleting dose. And this was for ethical reasons. We didn't think we could take a healthy HIV-positive person and take them to a transplant with potentially lethal effects of the busulfan. This shows you schematically what it was. So it was a complicated study that occurred in basically 4 phases at the sites, either the local site for the patient or the research subject, and then City of Hope. Here the patient was screened, and primarily the screening delay that we had was to complete the tropism assays, which take about 2 months to complete. So we would then have the patient selected, come to here for mobilization, again with GCSF and Plerixafor. We collected the cells, manufactured them, while the patient went back to his home. And then he came here for a transplant with a caregiver, and he had to stay at City of Hope for 21 days. And we had this was totally outpatient. We had outpatient facilities for lodging. And he was seen each day for complications or whatever was needed. And went home when he had engrafted, that his white cells had hit a certain level. And then, if he got his CD4 counts up to 450, and if the viremia was still negative, and if we could detect disrupted CCR5 in those CD4 cells, he could begin analytical treatment interruption. And that's where we are right now. So let's go through some of the results. We screened 71 patients. 80% of them were screen failures. We were surprised at this, but it turns out 40% of all of our patients had an HIV that was not only R5. It was usually dual tropism, but some of them were X4 virus. We were surprised at that, but that's what we saw. The next leading cause of dropout was that they actually got into the procedure and said, wait a minute, this is not your father's HIV trial. This is something totally new. And so we had some patients drop out when they got the first couple doses of GCSF and that sort of thing. The reasons given, the time commitment, number one. An AIDS doctor suggested, are you crazy? You should drop out of this trial. And 3 of them for unknown reasons. But we did complete the trial, and we have treated 8 patients, and we then declared that the trial was then close to accrual. And this is what we've seen. All the patients engrafted beautifully. In red is the platelets, in blue is the white blood cell count, and in green is the absolute neutrophil count, which tends to be the most important thing. And you can see that with busulfan, you tend to nadir around day 13 or so, 13 or 14. And this is cohort 1 that got the low dose busulfan. This is cohort 2 that got the higher dose. And they all engrafted then by day 15 in cohort 1, by day 14 in cohort 2. The one thing different between these 2 patients is the clinicians who do outpatient autologous transplants tend to use GCSF when they nadir, because they like to get them back to a white count that they don't have to worry about. But we were noticing that we were seeing increase in the marking, that is the genetic disruption in the peripheral blood in association with GCSF. So we said we don't want to remobilize cells that have already engrafted. So we stopped the GCSF, and in fact, these 2 patients engrafted very nicely and did not really have, except for the peak, they did not have any prolonged period of time to engraftment. So we would recommend not using GCSF. We don't know whether it affected the engraftment or not. Is it safe? It was very safe. If you look at this slide from the right to the left, you'll see the grade 4, grade 3, grade 2. No grade 3 or 4 during the busulfan period. There were some grade 1 and 2 problems, fatigue, constipation, things like that. But nothing serious. There were no serious adverse events. This is between the day of infusion and day 30. Again, very few problems. Again, those that we did see tended to be due to the high-dose busulfan. Mucositis was seen in 3 of the patients at grade 3. And that is a real problem. 1 patient had to be admitted for a few days, actually. But the rest was with blood test abnormalities, mostly lymphocyte problems, white cell problems that were grade 4. Hypophosphatemia was seen in a couple of patients. But nobody had any serious adverse events. Again, day 31 to day 100, very similar things, no toxicity. So the research outcomes then, they want to go over it. This is the recovery of CD4 counts. Relatively slow recovery of CD4 counts, and it tends to look like with the high-dose busulfan in green, the CD4 counts have recovered slower than the lower-dose busulfan. The percentage of CD4 cells, however, has been stable. And if you came in between 20 and 25, you tended to stay there. If you came in higher, you tended to stay there. And this is what you see if you were to look at patients who have been transplanted, again, autologously, for AIDS lymphoma. If you look at here, here's chemotherapy, and then you get to your transplant, your autologous stem cell transplant, and you have low levels of CD4s. And they go down with the collection of cells that you need for the transplant. And then they slowly rise. And so by 1 year, they're still not up to 400 mean. By year 3, they're just hitting above 400. So it does take a while for CD4s to recover. And so we don't think that was a specific problem with our regimen. Now this shows the key part, the CCR5 disruption in peripheral blood. Again, the green is the high-dose busulfan. We're seeing better disruption. Now this is presented as the number of reads in a MiSeq assay. So you're looking actually at the disrupted sequence as a percentage of total reads. So these can be thought of as the percentage of your alleles that are disrupted. And you can see that we're in the range of about 1% to 3%, maybe 4% early on. And then it levels out. We were pleased to see that we could take cells through this very rigorous process, get them back into the patient, and have their progeny still producing some disrupted cells, even out past between 1 year and 2 years. We were not happy with the fact that it's a very low number. Obviously, 2 to 3% of your total alleles is not sufficient to be protective ultimately. So we asked the question, what is it about this that we might want to change in the future? That is, did we do something wrong with the busulfan? That is the half-life in patients so different that we did not hit the targets we wanted? Was the zinc finger disruption an abnormality? That is, did we really have low efficiency? And what is the nature, the composition of the stem cells you actually get through this process and infuse? Are they really stem cells? So we looked at that, and in fact, we did hit our targets for busulfan, right on the nail, really, at least for the 12,000. But admittedly, 22,000 would have been better, but obviously we can't do that. So we need better conditioning. The zinc finger disruption was a surprise to us, because we started off really good with the first patient, and then we got really bad. So we closed the study when the third patient only had 8% disruption. And we did a root cause analysis. We looked at everything. We looked at the messenger RNA that was still at the factory in Germany. We looked at the messenger RNA that had never been sent to us, but it was being held at Sangamo. And we looked at our own freezers to see if we were losing the potency of the messenger RNA. And there was no difference in any of those three things. The one thing that we did find, we had to increase the amount of messenger RNA from 75 micrograms per mil to 150. So at 150, we then returned to the ability to meet our previously seen knockout of the disruption of the CCR5. So that's how we finished the trial at 150 micrograms per mil of messenger RNA in the electroporation cell. The cell yields were very high. As I said, we started with like 1.5 to 2 billion cells. But we then lose them because this method is very inefficient. We select the cells, we lose right there 50 to 30% of the cells in the selection system. And then you deliver those cells to the electroporator and you lose some more. So the final overall recovery ranged from 25% of the total cells you start with to about 50%. But they are all viable and they are really good CD34 cells. And so we were infusing close to 10 million cells per kilogram, where our goal was to hit 2 million per kilogram. So that was good news. So the feasibility of the method is there. The efficiency remains to be improved. So we then said, well, what about the trip from the freezer to the bedside? Maybe something goes wrong there. So we looked at the viability of the cells at the end of the infusion. We got the last few drops of cells, and in fact, there's no loss of viability. And we always think that when the cell is thawed in DMSO, we always infuse them within minutes, if possible. Well, in this case, we often went 15 minutes to 30 minutes based on the requirements of the FDA. Still no change in viability, though, so that's not a problem. What about the stemness of the final product? Did you actually get into your bag of cells that are very, let's say, primitive and can serve as progenitors? Well, this is the gating strategy. So what you do is you start with CD34 cells, and you try to take lineage negative cells out, and you look only at the CD34 negative cells, and then you look for negative CD45RA and CD38 negative, and then you want CD133 positive, and then you would like to see CD90 positive, CD49F positive. We call those really primitive hematopoietic cells. If you could transplant these, you might be better off. And in fact, there's a paper from Hans Peter's group recently in which they did just that in monkeys. But this shows you that we actually do have these cells in our product. And then we looked at other products, and in fact, most of the cells are these Lin-negative CD34 cells. They don't make it all the way through. Whoops! They don't make it all the way through that staging area. But we do have about 1 to 15% of the cells that are the Lin-negative, CD45 negative, and CD38 negative cells. Well, what about the ones that actually make it to very primitive status, let's say CD90? And we do have those, again, very small numbers. But here we see that, in fact, you do have approximately what you'd expect to see in a normal distribution of mobilized hematopoietic cells. So we don't think that's a problem, although, of course, you want to increase those numbers, and you want to target those cells really for the zinc finger manipulation, and we don't know, in fact, what percentage of those actually had disruption of the CCR5 gene. So in conclusion, let me just say, in summary, we have the preliminary data for this. We've safely done 8 patients. We are looking at the reservoir now. We've sent the specimens at 12 months, at baseline and 12 months, apheresis materials to Rafiq Sekali, who's doing reservoir analysis. We're looking at T-cell immunological assay and recovery post-transplant, and the patients are in a ATI phase right now. What's the summary for the community? Why is it important? The primary reason is this, as I said, is the first time this has ever been done. We can actually show that you can take patients through a system like this, and you can take the cells through an electroporation like this and get them to re-engraft and express progeny that have the genetic manipulation. The results indicate engraftment. The studies on the HIV reservoir, as I said, are pending. How is this related to a cure? Well, it shows that zinc finger genome editing of blood stem cells to make resistant CD34s remains viable. We don't have it optimized, but it certainly remains viable. Now, should we be excited? I think we should be really excited about the fact that this was actually done. And it was a tremendous effort, at great expense to the subjects, to their caregivers, and to the sponsors in general. But it did work. We showed the test of concept. You can make cells, you can genome edit stem cells and see the effect in their progeny. We have to be cautious, though, because the technology is needed to improve this so that we can actually make it an effective part of cure strategy. So I want to thank all those that were involved and acknowledge them, primarily our team at City of Hope, the clinical team that involved not only our transplanters under Rita Krishnan, but Jayla Azari in San Francisco, Ron Michiasu, Tony Mills in LA, the funding group, Sangamo, and most importantly, the research subjects and the caregivers who really committed a lot of time to this work. Thank you.
Thank you so much, John. This is really an enormous accomplishment. I know how much work, patience it takes to do these clinical studies. Thank you very much. We've got time for 2 questions.
Yeah. Thank you. What are the... Did ART restart the criteria?
To restart the ART?
Yeah.
That's a good question. As I said, to start the ATI, you must have a certain level of cells, CD4 cells. You must have the marking in those cells. It must be aviremic. We then follow the HIV level. If it hits 5,000, then we start following it every week, and we stop at 50,000. Or the CD4 cells are followed, and if the CD4 cells fall below 350, then we follow every week. And if they do that 3 weeks in a row, we stop. Or if the CD4s hit 200 at any one time, we stop. Or if the HIV hits more than 50,000, I'm sorry, more than 100,000 at any one time, we stop. So that's.
Hey, Larry, sorry I beat you to it. So really quick, I'm fascinated by the CCR5 editing platforms because it feels like unless it's 100% edited, the therapies will not work. So with a platform like this, is there a percentage threshold that you think is going to be sufficient to cure these patients? Because unless it's 100%, the virus is going to just continue.
That's similar to Monique's question last time. I think that's a very good question. I think that when we were initially doing the zinc finger, we felt confident that you could get 2/3 of the cells homozygously knocked out. That still left the other third. It was heterozygote, and we said, well, maybe that's good enough. But of course, you have to get the frequency of the altered numbers up to high levels. Now we don't know what that is. Hans Peter's got a paper in monkeys that shows that you may need only 30% of the cells to be genetically modified for a protective effect, because there's some kind of a bystander effect or other T cells kick in and the immune system becomes healthy in the absence of a robust HIV infection. So we don't know the answer completely, but I don't think you need 100% of cells to have biallelic knockout of the gene of interest.
Yeah, I agree with that. I don't think you need 100%. We've shown that actually having some protection will facilitate hepatitis cells to really boost the immune response against HIV. Now, one quick question for Larry. Is that possible?
Yeah, it's quick. It's a corollary of Semi's question. So as you follow these people over time, what is the relative percentages of double knockout, homozygous CCR5, heterozygous CCR5, and quote,
Yeah, that's a difficult.
What's happening to the slope here?
Yeah, we don't know the answer to that in these patients. We know that in the preliminary work, the preclinical work, you're able to do that much easier on cloned cells. It is about 2/3 will be homozygous knockout. And as you improve the method, you may actually increase that. But at the present time, that's what we were expecting to see.
Okay. Thank you very much again, John.
Thank you.